Welcome to Biomania Revision Podcast. Today, we are starting very, very important unit that is genetics. So focus should 2x, now just put headphones and close your eyes, listen to the podcast and visualize in your mind.
That's right. We're about to decode, the very blueprint of life itself. We're diving straight into Unit 7 and specifically Chapter 4, Principles of Inheritance and Variation.
And this is so much more than just cramming facts for an exam. This is about understanding the code that literally builds everything alive on this planet.
It really is.
Yeah.
We're going on a journey today. We're starting with macroscopic things like pea plants in a garden, and we're going all the way down to the microscopic. You could call it a dance of chromosomes inside a cell.
Exactly. And we're tackling some of those huge questions, the ones that sound almost childishly simple until you actually try to explain them. I mean, have you ever stopped to really properly think about why an elephant always, without fail, gives birth to a baby elephant?
And not a baby giraffe or, a mouse. It seems obvious.
It seems completely obvious, but when you zoom in, that consistency, that fidelity is a biological miracle.
It is. Or another classic example, a mango seed. You plant it and you get a mango plant every single time. There's a rigid microscopic mechanism that ensures that continuity.
So that's what scientists mean by species breed true.
Precisely. An oak tree isn't going to accidentally sprout a pine cone. There is a script, a very detailed script, that's being followed with incredible accuracy.
And this is the big but. It's not just a photocopy machine, is it?
No, not at all.
Because you look at siblings, I mean, they come from the exact same parents, the same, source code, and yet they're different.
They can be incredibly different, or sometimes remarkably similar. That's the whole puzzle.
And that gets us to the core of what this is all about, right? The two big ideas.
Exactly. You've hit on the dual nature of genetics. On one side, you have inheritance. That's the process, the mechanism of passing traits from parent to progeny. That's the stability part.
The part that makes elephants have elephants.
Right. But then you have the other side of the coin, variation. And that's the degree by which that progeny differs from its parents and, from its siblings.
And what's wild is that humans have had an intuitive grasp of this for a very long time. I mean, way, way before anyone knew what DNA was.
Oh, for thousands of years. If you go back to, say, 8,000 to 1,000 BC, our ancestors had already figured out that the secret to variation was hidden somewhere in sexual reproduction.
They didn't know the science, but they knew the results.
They knew the results. They knew if you took a wild animal that had a trait you liked, maybe it was more docile or it produced more milk, and you selectively bred it, you could amplify that trait. You could keep it.
That's where something like the Sahiwal cows in Punjab come from. That's the textbook example, isn't it?
It's the perfect example. The Sahiwal cows we see today were developed over a long, long time through artificial selection, through domestication from ancestral wild cows.
So the breeders in Punjab, they knew that it worked. They knew that traits were inherited.
Yes. They knew that if they picked the very best bull and the very best cow, they'd probably get a better calf. But they had absolutely no idea how it worked or why. They were operating completely in the dark, really, just using trial and error.
And that's the perfect setup for our main character to enter the story, the person who finally switched on the lights.
The man, the monk, Gregor Mendel.
The father of genetics. And it's so important to remember, he wasn't just some guy who liked gardening.
No, he was a trained scientist wearing a monk's robe. He had a deep understanding of mathematics and the scientific method.
Okay, so let's set the scene for everyone. We're talking about the mid-19th century. There's a really specific window of time here, 1856 to 1863.
7 years. For 7 straight years, Mendel is conducting these hybridization experiments in the garden of his monastery in Brno, which is now in the Czech Republic.
And his approach was, well, it was totally alien to what biology was at the time.
Right, completely. At that point, biology was mostly a descriptive science. It was about observing, drawing, and categorizing. You'd draw the details on a leaf or describe the behavior of an animal.
And Mendel comes along and brings math.
He brought math, and that changed everything. He was the first person to apply statistical analysis and mathematical logic to biological problems.
Which, as we'll get to, is a big part of the reason people just ignored him at first. They thought, what is this? This isn't biology.
It was a massive paradigm shift, but it was absolutely necessary. You see, he didn't just say some of the plants in the next generation were tall. He counted them. He counted exactly how many were tall and how many were dwarf.
So he had a huge sample size.
A very large sampling size, which gave his data a credibility that no one had ever had before. It's like if you flip a coin 10 times, you might get 7 heads and 3 tails and think something's weird with the coin.
Right.
But if you flip it 10,000 times, you're going to get very, very close to that 50-50 split.
Yep.
Mendel's work was on that scale. He worked with thousands of plants.
And his subject was the garden pea, scientific name Pisum sativum. But he was very specific about the peas he used. He didn't just plant any old seeds.
No, this is probably the most critical part of his experimental design. He used what he called true breeding lines.
Okay, that's a key term. Let's make sure we all have that locked in. What exactly is a true breeding line?
A true breeding line is a plant that has undergone continuous self-pollination for several generations and shows stable inheritance for a specific trait.
So let's say you have a tall plant. You let it pollinate itself. You collect the seeds. You plant them.
And if all the offspring, all the babies, are also tall,
You're on the right track.
you're on the right track. Then you take those plants, let them self-pollinate, and if all the grandbabies are also tall, and so on and so on, eventually you can say with confidence that you have a true breeding line. It's genetically pure for that trait.
Okay, focus mode, everyone. Let's close our eyes and actually visualize Mendel's garden. He didn't just pick 1 or 2 things to look at. He carefully selected 14 true breeding pea plant varieties.
Which, in reality, were 7 pairs of contrasting traits.
Exactly. And you need to know these 7 traits cold, so let's list them. I want everyone listening to actually picture them. Let's build the plant from the ground up.
Good idea. So, starting from the very bottom, or, well, the overall size.
Stem height. This one is the easiest to remember. It's either tall or dwarf. Picture a tall, climbing vine next to a small, stout bush.
Simple enough. Next, let's move to the flower. He looked at 2 different traits on the flower. First one is flower color.
And the options were violet or white.
Correct, with violet being the one that turned out to be dominant. The second flower trait was flower position.
Okay, axial versus terminal. These words might be a little tricky. What do they mean visually?
Axial means the flowers grow along the stem, coming out of the axil. That's the little junction where a leaf stalk meets the main stem. So they're all along the sides.
Got it. And terminal.
Terminal means all the flowers are bunched up right at the very top, at the terminus or the tip of the plant.
Okay, so we've got the stem, we've got the flower. Next up is the pod, which is the fruit of the pea plant. 2 traits here as well.
First is pod shape. This could be either inflated or constricted.
Inflated is easy to picture. It's just a full, smooth, rounded pod.
Right, like it's been inflated with air. Constricted is the opposite. It's sort of bumpy and shrunken in, where you can see the outlines of the peas inside.
Okay, and the second pod trait is pod color, green versus yellow.
And everyone, put a big mental star next to this one. This is a classic trap in exams. For the POD, the dominant color is green.
Green is dominant for the pod. Okay, now let's crack open that pod and look at the actual seeds inside. Again, 2 traits.
First, seed shape. This one's pretty straightforward. Round versus wrinkled.
And finally, seed color. And this is the other half of the trap, isn't it?
This is it. The options are yellow versus green, but for the seed, the dominant color is yellow.
So let's say that one more time to lock it in. For the pod, green is dominant. For the seed inside, yellow is dominant.
Right. The best way to visualize it is to picture the thing you'd actually buy at the store. A dominant green pea pod that you crack open to find dominant yellow seeds inside. Well, if you were buying split peas anyway.
Perfect. So Mendel has his pure lines. He has his 7 pairs of contrasting traits. Now the real work begins. He starts crossing them. And this brings us to the first big concept, the inheritance of one gene.
Or what we call a monohybrid cross.
Mono meaning one. So he's just looking at one single trait at a time?
Exactly. Let's stick with the simplest one, stem height. He takes a true breeding tall plant and he crosses it with a true breeding dwarf plant. This first generation is called the parental generation, or the pea generation.
Now, a practical question. Peas normally pollinate themselves. How does he force them to cross-pollinate?
This is where the painstaking work comes in. He had to perform a process called emasculation.
Sounds intense.
It's very delicate. He would have to carefully open up the flower bud of the plant he chose as the female parent, and, using tiny forceps, remove the anthers, the male parts, before they had a chance to mature and shed their pollen.
So he's preventing self-pollination.
Exactly. Then he would collect pollen from the male parent plant and carefully dust it onto the stigma of the emasculated flower. It was incredibly tedious, precise work.
Wow. So he does this. He collects the seeds from that cross, plants them, and waits. The plants that grow are the first hybrid generation. We call it the F1 generation, which stands for filial 1.
And what he sees is astonishing.
Every single plant is tall.
Every single one. There were 0 dwarf plants. And this is just as important, there were no medium height plants.
Which is really weird, right? You'd think if you cross a tall thing and a short thing, you'd get something in the middle.
That's what the prevailing idea of blending inheritance would have predicted, like mixing black paint and white paint to get gray. But that's not what he saw. The dwarf trait seemed to have just vanished completely.
And he saw this same pattern for all 7 traits, didn't he?
Yes, he'd cross a violet flower with a white one, and all the F1s were violet. He'd cross round seeds with wrinkled seeds, and all the F1s were round. One of the parental traits was always expressed, and the other was always hidden.
But of course he didn't stop there. This is where it gets really clever. He took these F1 tall plants and he allowed them to self-pollinate.
He essentially let them breed with themselves. This produced the next generation, the F2 generation. And this is where the real magic happened.
The dwarf trait came back.
It reappeared, as if from nowhere.
Like a ghost in the machine.
It was. And because he was a mathematician, he counted. And he found that in the F2 generation, about 1/4 of the plants were dwarfs, and the other 3/4 were tall.
So a ratio of 3 tall to 1 dwarf.
A consistent 3-to-1 ratio. And again, crucially, no blending. The tall F2 plants were just as tall as their tall grandparent, and the dwarf F2 plants were just as dwarf as their dwarf grandparent. No medium heights.
Okay, so Mendel is looking at this data. He doesn't know about genes or DNA or anything. What does he conclude from this? What did he call the things that were controlling these traits?
He called them factors. A very simple, elegant name. He proposed that something was being stately passed down, completely unchanged, from parent to offspring through the gametes.
And today we call those factors genes.
We call them genes. Genes are the fundamental units of inheritance. They contain the information needed to express a particular trait.
And the different versions of a gene, like the tall version and the dwarf version, are called alleles.
That's the word. Alleles are just slightly different forms of the same gene. I like your ice cream analogy. If the gene is ice cream flavor, then the alleles are chocolate and vanilla. For the height gene, the alleles are tall and dwarf.
And we use letters to represent them, right? A capital letter for the dominant trait.
And a lowercase letter for the recessive one. So for height, we'd use a capital T for the tall allele and a lowercase t for the dwarf allele.
Okay, let's nail down some more of this terminology. It's revision time, after all. Every organism gets one factor from each parent, so they have a pair of alleles for each gene. What do we call it when the 2 alleles are identical, like big T, big T?
That's homozygous. Homo means same. So TT is homozygous dominant, and PT is homozygous recessive. These are the true breeders Mendel started with.
And what if the alleles are different, like big T, small t?
That's heterozygous. Hetero means different. This is the genetic makeup of the F1 generation.
Got it. And that leads to the crucial difference between how something looks on the outside versus what its genes are on the inside.
Yes, and these 2 words are non-negotiable. You have to know them. The observable physical characteristic, tall or dwarf, is the phenotype.
Phenotype, physical. That's a good way to remember it.
And the actual genetic makeup, the combination of alleles like TTT to your TTP, is the genotype.
Okay. So Mendel realized that in that F1 hybrid, the TT plant, its phenotype was tall. It looked exactly the same as the TT homozygous parent.
Right. From this, he proposed that in a dissimilar pair of factors, or a heterozygous genotype, one factor dominates or masks the effect of the other.
And that's the concept of dominance. The T allele is dominant over the T allele, and the T allele is recessive.
A recessive trait is only expressed in the phenotype when the dominant allele is absent, so only in the homozygous TKA genotype.
This is all starting to click into a logical system. Now, to really visualize this and predict the outcomes of these crosses, there's a tool that every biology student learns.
Ah, yes. The Punnett square, developed by a British geneticist, Reginald C. Punnett. It's a simple graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross.
So let's walk through it for that F2 cross. We're self-pollinating an F1 plant, so the cross is TTX-TT.
Okay, so first you draw a square and divide it into 4 smaller boxes. Now we need to figure out the gametes that each parent can produce.
During gamete formation meiosis, the 2 alleles in the parent separate. So a T parent will produce 2 types of gametes.
Exactly. 50% of its gametes, say the pollen, will carry the T allele, and the other 50% will carry the T allele. The same goes for the eggs.
So along the top of our Punnett square, we write the two possible gametes from one parent, T and T. And down the left side, we write the two possible gametes from the other parent, T and T.
Perfect. Now you just fill in the boxes, combine the letter from the top with the letter from the side. The top left box gets a T from the top and a T from the side. So the genotype is TT.
The top right box gets a T from the top and a T from the side. So TT.
Bottom left box gets a T from the top and a T from the side, also TT.
And the bottom right box gets a T from the top and a T from the side, so TT.
And there it is. You can see the result right in front of you. You have one TT square, two TT squares, and one T square.
Which gives us our genotypic ratio of 1.2.1. One homozygous dominant to two heterozygous to one homozygous recessive.
But now let's look at the phenotype. What do they look like? The TT plant is tall. The two TT plants are
also tall because T is dominant.
Right. And the 1T plant is dwarf. So you have 3 tall plants and 1 dwarf plant.
And there's our phenotypic ratio of 3.1. The math works.
The math works perfectly.
Yes.
And the source material points out that you can even represent this with a binomial expression, like from algebra class.
Right. AX plus by 2, How does that connect?
Well, think of the gametes from the F1 generation as having a 50% or 12 chance of being T and a 12 chance of being T. So the expression is $12T plus 12T22.
Ah, so you're squaring it because you're combining it with itself.
Exactly. And when you expand that algebraically, you get $12T2 plus 22 plus 22s plus 2 plus 12T22, which simplifies to
$14 TT plus 12 TT plus $14, the exact same ratio.
It just proves that this isn't some biological magic. It's pure probability at work.
Oh, okay. So this Punnett square is powerful, but Mendel faced a practical problem. Let's say you're out in the garden, you see a tall pea plant. Your phenotype is tall, but what's its genotype?
You can't tell just by looking. Is it homozygous dominant TT or is it heterozygous TT? They look identical.
This is a classic genetics problem. How do you figure out the genotype of an organism showing a dominant trait?
Mendel devised a brilliant solution for this. It's called the test cross.
A test cross. Okay, what are we testing and how does it work?
We are testing the unknown genotype of that tall plant. And the method is to cross the mystery plant with a plant that is homozygous recessive. In this case, a dwarf plant, genotype TT.
Okay, stop there. Why specifically a recessive parent? Why not cross it with another tall one?
That's the key. The recessive parent is, you could say, genetically clean. It only has the recessive T allele to contribute to its offspring. It can't mask anything. It essentially provides a blank slate against which the alleles of the unknown parent can reveal themselves.
Okay, that makes sense. So let's visualize the two possible outcomes. Scenario A. Our mystery tall plant is actually homozygous dominant TT.
Okay, so we're crossing TT with TT. The TP parent can only make one type of gamete, carrying a T. The T parent can only make gametes carrying a T.
So 100% of the offspring will have the genotype TT.
And what will their phenotype be?
All of them will be tall.
So there's your answer. If you perform the test cross and all of your offspring show the dominant phenotype, your mystery parent was homozygous dominant.
Okay. Now for scenario B, our mystery tall plant is heterozygous TT.
Right, so now we're crossing TT with TT. The heterozygous parent produces 2 types of gametes, remember, half get T, half get T.
And the recessive parent still only gives T.
So half the time, a T gamete will meet a T gamete, making a T offspring. That one will be tall.
And the other half of the time, a T gamete will meet a T gamete, making T offspring. And that one will be dwarf.
Exactly. You get a 1.1 ratio, 50% tall, 50% dwarf. So the rule is, if even a single recessive phenotype, a single dwarf plant, appears in the offspring of your test cross, you know for certain your mystery parent was heterozygous.
Simple, logical, and just. Brilliant. Okay, so based on all this work from the monohybrid cross, Mendel formulates his first 2 fundamental laws of inheritance.
Yes, let's state them very clearly for everyone revising. The first is the law of dominance.
And it really has 3 parts.
It does. First, characters are controlled by those discrete units he called factors. Second, these factors occur in pairs. And third, in a dissimilar pair of factors, a heterozygous individual, one member of the pair dominates the other.
The dominant allele masks the recessive one.
Right. And this law explains why that F1 generation was uniform, all tall, and why the F2 generation showed that 3.1 ratio.
Okay, that's law number 1. What's the second?
The second is the law of segregation. This law is based on the critical observation that the alleles do not blend or mix in any way. Both parental traits are recovered, completely unchanged, in the F2 generation.
The dwarf trait wasn't diluted. It was just hidden and came back perfectly dwarf.
Exactly. So the law states that during gamete formation, The factors or alleles of a pair segregate. They separate from each other so that a gamete receives only one of the two factors.
It's like a rule for packing your bags for a trip. You have two shirts, a red one and a blue one, but your suitcase, the gamete, only has room for one. You have to pick.
That's a perfect analogy. You can't take both. And this law is considered universal. For diploid organisms making gametes, this segregation happens. A homozygous parent, TT, produces only one kind of gamete, all T. A heterozygous parent, TT, produces two kinds of gametes, T and T, in equal proportions.
Okay, so that is the beautiful, clean, predictable world of Mendelian genetics.
The baseline, the perfect world scenario.
But as we know, nature is rarely that simple. It loves to throw in exceptions and complications. Mendel was actually very lucky with the traits he chose in peas.
He was incredibly fortunate. They all showed clear dominance. But as scientists looked at other organisms, they quickly found cases that didn't fit these neat rules.
And that brings us to the fascinating world of non-Mendelian inheritance, the deviations from the plan.
Right, and the first one we absolutely have to visualize is called incomplete dominance.
For this one, we need to switch plants. Forget the peas for a moment. Picture a flower called a snapdragon, or its scientific name, Antirrhinum.
Okay, so remember in peas, a dominant red crossed with a recessive white would give you an F1 that was all red. Simple dominance.
Right.
But in snapdragons, if you take a true-breeding red-flowered plant, let's call it RR, and cross it with a true-breeding white-flowered plant, RR, the F1 generation is not red.
It's pink. The genotype is R, and the phenotype is pink.
Pink. It doesn't resemble either parent. It's an intermediate phenotype. It looks, for all the world, like the blending that Mendel disproved.
But it's not actually blending, is it?
It is not. And we know this because of what happens in the F2 generation, when they took those pink F1s and self-pollinated them.
They didn't just get more pink flowers.
No, the original parent colors came back. They got a ratio of 1 red, 2 pink, 1 white.
Wait a second. That 1.2.1 ratio, that's the same genotypic ratio we saw in Mendel's cross, 1 RR, 2 RR on R.
Precisely. The underlying mechanism of segregation is still perfectly Mendelian. The alleles are still separating cleanly. What's changed is how the genotype translates to the phenotype.
So instead of the 3.1 phenotypic ratio, the phenotypic ratio has now become 1.2.1, exactly matching the genotype.
Right. The heterozygote, RR, has its own unique phenotype. The pink color allows us to physically see the heterozygous state.
Okay, so why does this happen? The source material goes into a really cool molecular explanation for dominance using enzymes. Can we unpack that? Like, what does it even mean for an allele to be recessive?
This is such a critical concept to grasp. A gene, fundamentally, is a piece of code. It contains the information to express a trait. And very often it does this by telling the cell how to build a specific enzyme.
An enzyme that does a job.
An enzyme that catalyzes a reaction. So let's imagine the normal allele, the unmodified one, produces a fully functional enzyme. And this enzyme takes some substrate molecule and converts it into, say, a red pigment.
Okay, so normal allele working enzyme, red color. Simple.
Now, imagine a mutation happens. The allele gets modified. It becomes the recessive allele. This modified code could result in a few different outcomes.
Like what?
1, it might produce a slightly less efficient enzyme. 2, it might produce a completely non-functional enzyme. Or 3, it might produce no enzyme at all.
So in options 2 and 3, no pigment gets made. And if there's no red pigment, the flower looks white.
Exactly. Now think about the heterozygote, the pink flower. It has one normal allele and one modified non-functional allele.
So it has only half the instruction manual.
Right. That one normal allele produces some of the functional enzyme. It's doing the job. But maybe it can't produce enough enzyme to make the flower fully deeply red. It produces enough for a pink color.
So incomplete dominance is like a dosage effect. One dose of the good allele isn't enough to get the full effect.
That's a great way to think about it. The unmodified allele still dominates in the sense that it's the one doing the work, but it's not strong enough to completely mask the fact that the other allele isn't contributing at all.
That makes so much more sense. Recessive isn't some active force, it's usually just a broken or missing function.
In most cases, yes. That's exactly right.
Okay, let's move to the next deviation. This one is called codominance.
Codominance. So if incomplete dominance was a mix, like pink, codominance is more like pinga. They're both on the throne at the same time. They're co-rulers.
And the best example of this is human ABO blood grouping.
The classic example. We all know about blood types, A, B, and O. This whole system is controlled by a single gene, which we call gene I.
So what is this gene actually
Well,
doing?
the plasma membrane of our red blood cells has these sugar polymers that stick out from the surface. The kind of sugar that gets put there is controlled by this gene I.
And for this gene, there are three different alleles in the human population, right?
Correct. There's allele IOEA, allele AVIL, and allele ADALR.
Hold on, three alleles. I thought we only get two alleles for every gene, one from each parent.
You do. Any single individual can only have a maximum of 2 of these alleles. But when you look at the entire human population, there are 3 different versions of this gene floating around in the gene pool. This phenomenon is called multiple alleles.
Got it. So an individual has two, but the population has more. So what do these three alleles do?
IA and IBI both code for enzymes that produce a slightly different kind of sugar on the cell surface. The allele lighter, on the other hand, doesn't produce any functional enzymes.
So it produces no sugar.
It produces no sugar. That makes it recessive. So I allele is dominant over I and ALR. If your genotype is AALE, your blood type is A.
And same for B. IL is dominant over I dollars, so IABA gives you type B blood.
Correct. But here's the crucial part. What happens when a person inherits both an I allele and an IBL allele?
That's the codominant moment.
That's it. They don't blend to make some intermediate sugar. They don't mask each other. They both get fully expressed. The red blood cell ends up with both A-type sugars and B-type sugars sticking out from its surface.
And that person's blood type is AB.
Codominance. Both alleles contributing equally and fully to the phenotype.
Wow. So let's quickly do the math on that. With three alleles, and I allow how many different genotypes are possible.
Let's list them out. You can be homozygous for A, IAA, IAA, or heterozygous, IAO, that's 2. You can be homozygous for B, IAB, IAB, or heterozygous, IABAA, has another 2. You can be codominant AB, IAAABD, and you can be homozygous recessive for O.
So 6 possible genotypes?
6 genotypes, but they only produce 4 different phenotypes: type A, type B, type AB, and type O.
Okay, there's one more really tricky, subtle deviation mentioned in the source material. It's about how dominance itself can be context-dependent. It brings up the idea of pleiotropy.
Yes, this is a fantastic example that shows how nuanced biology can be. Pleiotropy is when a single gene influences multiple, seemingly unrelated phenotypic traits.
And the example is starch synthesis in pea seeds, the very same plants Mendel studied.
The very same. So there's a single gene that controls starch synthesis. Let's call it alleles B and B. Plants with the genotype BV are very efficient. They produce a lot of starch, and as a result, they have large starch grains.
Okay. And plants with the genotype Bob are less efficient, produce less starch, and have smaller starch grains.
Right. Now, what about the heterozygote Beeb?
Well, based on what we just learned about incomplete dominance, I'm going to guess it produces an intermediate amount of starch.
You're exactly right. BD plants have intermediate-sized starch grains. So if the phenotype you're measuring is starch grain size, this gene shows incomplete dominance.
Okay, but wait. This same gene also affects the overall seed shape. All that starch makes the seed plump and round. Less starch makes it shrivel up as it dries.
Precisely. So BB seeds, with their large starch grains, are round. Yeah. And BB seeds, with their small grains, are wrinkled.
What about the bead seeds? They have an intermediate amount of starch, but what do they look like?
They look round. The intermediate amount of starch is still enough to prevent the seed from wrinkling.
Whoa. So if the phenotype you're looking at is seed shape, the B allele is completely dominant over the B allele.
And there's the punchline. The same gene, the same 2 alleles. But depending on which phenotypic level you choose to examine, the microscopic grain size or the macroscopic seed shape, it exhibits a different pattern of dominance.
That's amazing. It really drives home the point that dominance isn't some intrinsic absolute property of an allele.
It's not an autonomous feature of a gene. It's a relationship between alleles, and it depends on the gene product and the specific phenotype you're observing.
My mind is officially a little bit blown. Okay, we have conquered the inheritance of one gene and its exceptions. Mendel, however, was not finished. He leveled up.
He did. He moved on to the inheritance of 2 genes, the dihybrid cross.
He wanted to know if different traits were inherited as a package deal or if they were independent.
Exactly. Do seed shape and seed color affect each other? To find out, he set up a cross between a pea plant that had round yellow seeds and one that had wrinkled green seeds.
Let's define the genotypes. Round is R, wrinkled is R, yellow is Y, green is Y. So he's crossing a true breeding RRYY plant
With a true breeding rye plant.
The RRY parent can only make one kind of gamete, RY, and the Becker parent can only make one kind of gamete.
So when they cross, the F1 generation is straightforward. They all have the genotype RRYY.
And what's their phenotype? What do they look like?
Well, R is dominant to R and Y is dominant to Y. So they are all round and yellow.
Okay, so that just confirms the dominance relationships. Now for the main event. He takes this F1 hybrid, RRYY, and he self-pollinates it.
And this is where the Punnett square explodes in size.
It's no longer a 2x2 grid. It's a 4x4 grid with 16 squares. Because first, we need to figure out all the possible gametes the RRY plant can make.
And to do this, we have to assume the two genes are sorting independently. The segregation of the R's is a separate event from the segregation of the Y's.
It's like flipping two different coins at the same time.
Exactly. So you can get a capital R with a capital Y, making an RY gamete. You can get a capital R with a lowercase y, making an Ry gamete.
You can get a lowercase r with capital Y for an RY gamete. Or a lowercase r with a lowercase y for an Ry gamete.
4 possible types of gametes in equal proportion. And you put those 4 across the top of the Punnett square and the same 4 down the side.
And when you fill in all 16 boxes and tally up the phenotypes, you get a very famous, very specific ratio.
The phenotypic ratio of 9.3.3.1.
Let's break down what those numbers mean. The 9 represents the plants that look like the dominant parent.
9 are round and yellow. The 1 at the very end represents the plants that look like the recessive parent.
1 is wrinkled and green.
But the 3 and the 3 in the middle, those are the interesting ones.
Those are the new combinations, the recombinants. They have a mix of parental traits.
You get 3 plants that are round but green, and 3 plants that are wrinkled but yellow.
The very existence of these new combinations was the proof Mendel needed for his third great idea.
The law of independent assortment.
And this law states?
It states that when 2 pairs of traits are combined in a hybrid, the segregation of one pair of characters is independent of the other pair of characters.
In other words, the gene for seed shape doesn't know or care what the gene for seed color is doing when the gametes are being made. They sort themselves out randomly.
They assort independently. Now, we should add a small caveat. Mendel was lucky again. The genes he happened to choose were either on different chromosomes or were very far apart on the same chromosome.
Which allowed them to assort independently.
Yes. If 2 genes are physically located very close together on the same chromosome, they tend to be inherited together. This is called gene linkage, which is a topic for another deep dive. But for Mendel's third law, the principle of independent assortment holds true.
Okay, so we've covered the 3 laws. An incredible body of work. Mendel publishes all of this in 1865. It's revolutionary. It's the key to heredity. And then crickets.
Complete silence. For 35 years, his work was essentially lost. It sat on library shelves, unread and unappreciated.
It's one of the great tragedies of scientific history. Why? Why did the world just completely ignore the man who had figured out the fundamental rules of life?
Well, there were a few key reasons. First, and this is a simple one, communication back then was terrible. There was no internet, no email, no global scientific journals. He published his findings in the proceedings of a local natural history society. It just didn't get wide circulation.
So not many people even saw it.
And the ones who did probably didn't understand it or didn't believe it. His core concept of these stable, discrete factors that didn't blend was completely radical.
What did people believe at the time?
The prevailing theory was one of blending inheritance and continuous variation. The idea was that traits flowed from one generation to the next like rivers merging. Mendel came along and said, no, it's not a river. It's made of tiny, hard particles that never change. It just didn't fit the worldview of his contemporaries.
And then there was the math problem.
Oh, yes. The math. Using mathematical and statistical logic to explain a biological phenomenon was, to many, just unacceptable. It was a foreign language. Biology was a descriptive science, not a quantitative one.
And the final piece of the puzzle, he couldn't actually show them a factor, could he?
That was the biggest hurdle. He had no physical proof. He couldn't put a gene under a microscope and say, there it is. He couldn't say what they were made of or where they were located in the cell. It was all inferred from his data. It was too abstract for most people to accept.
But then, at the turn of the century, the year 1900 arrived, and everything changed.
The golden year. 3 different scientists, working completely independently in 3 different countries, all stumbled upon Mendel's original paper while researching their own work on heredity.
And who were they?
Hugo de Vries, Carl Quarens, and Erich von Schermach. And they had all, through their own experiments, arrived at conclusions that were remarkably similar to Mendel's.
So they didn't just rediscover his work, they validated it.
They did, and crucially, in the 35 years that had passed, technology had caught up.
We had much better microscopes.
Much better microscopes. And in that time, cytology, the study of cells, had become a major field. Scientists had been observing what happens inside cells during division.
And they'd seen these mysterious thread-like structures in the nucleus that seemed to double and then divide just before the cell split in two.
They didn't know what they were.
Yeah.
But they took on dyes very well, so they named them chromosomes, which literally means colored bodies.
So you have the geneticists rediscovering Mendel's abstract laws over here, and the cytologist watching these physical chromosomes move around over there.
And they were two separate worlds. Until 1902, when two scientists, Walter Sutton and Theodore Boveri, put the two worlds together.
They were the ones who had the great aha moment. They looked at the behavior of chromosomes during meiosis, and they looked at Mendel's laws, and they realized
They're doing the same thing. The movement of chromosomes perfectly parallels the behavior of Mendel's factors.
And this became known as the chromosomal theory of inheritance.
Yes. Sutton and Boveri laid out the parallels very clearly. For example, Mendel said factors come in pairs. Well, chromosomes also come in homologous pairs.
And Mendel said the factors in a pair segregate during gamete formation.
And that's exactly what homologous chromosomes do during meiosis I. They separate.
Let's visualize that. Anaphase I of meiosis. Imagine you have two pairs of homologous chromosomes in a cell, maybe a long pair, one you got from your mom and one from your dad, and a short pair, also one from each parent.
Okay. When the cell prepares to divide to make gametes, these pairs line up at the center, at the metaphase plate. But how they line up is completely random.
So the long maternal chromosome might line up on the left side with the short paternal one?
Or it could line up with the short maternal one.
Yeah.
The orientation of the long pair has no influence on the orientation of the short pair,
They assort independently.
just like Mendel's genes for seed shape and seed color.
So Sutton put it all together. He argued that the pairing and separation of a pair of chromosomes would lead directly to the segregation of the pair of factors or genes that they carried.
The chromosomes were the physical vehicles for Mendel's abstract factors. The genes were passengers on the chromosomes.
And just like that, the physical basis of heredity was found. The gap was bridged.
It was a beautiful synthesis. It connected the abstract mathematics of a monk's garden to the physical, observable reality of the cell nucleus. The theory of inheritance finally had a home.
And that is a perfect place to wrap up this massive revision deep dive. What a journey. We've gone from a 19th century monastery garden right down into the nucleus of a cell.
We really have. We've connected the dots from simple observation to statistical laws and finally to physical structures. It's the foundation of everything we know about genetics today.
And understanding these principles, dominance, segregation, independent assortment, it's not just academic, it's the key that lets us predict things.
It's everything. It helps us understand the probability of genetic disorders, it lets us trace our own ancestry through our genes, and it provides the raw mechanism for evolution itself. This is the starting point for it all.
All right, learners, that is it for Chapter 4. Say like the video and tell how it was, and if you want more chapters to revise, then tell in comment section.
That's right. Keep questioning, keep visualizing those Punnett squares, and keep making those connections.
We'll see you in the next deep dive.